Welcome to the panel on genetic medicine. We've had a session like this pretty much every time we've had a GRI meeting, but today's a little bit different because in the past we've only been able to have researchers working on genetic medicines that are focused on GRIN disorders. But today we're lucky to have back Dr. Amy Ramsey, who is doing work on GRIN disorder, GRIN 1, but also Rick Huganir, who's doing work on GRIA2, and Dr. Geoff Swanson, who's doing genetic preclinical work on GRIC2. So welcome to all of you, and thanks for being part of today's session. For the format for today, we're going to just start with a couple of a bit of a dialogue on some of the points that are relevant. Then we're going to move into individual presentations from the speakers, and then finish with another discussion and Q&A. So the first thing that I wanted to ask the panel is that in each of the pieces of work, each of your studies involve the creation of a patient knock-in mouse, so an animal that mirrors, I guess, a patient variant. So tell us, maybe I can get Amy to start by telling us how that works and why that's helpful. Maybe we've lost Amy for a minute. Ricky, I think you've made more mice than anybody else. Why don't you start with it?
Sure. I mean, mouse models. I'm a great believer in mouse models. They don't model all diseases, obviously, or all even psychiatric, neurological, and psychiatric diseases. But what, in my opinion, they model intellectual disability um, and these monogenic autism-related genes. Um, that model very well in mice. So not it's not perfect, but what we found working on SYNGAP and GRIA, both AMPA receptor mutations, they model the intellectual disability, the autism, the social deficits, the seizures. So it's turned out to be really incredibly helpful for the development of therapeutics, which of course we'll get into that discussion shortly.
And Geoff, how do you find a mouse that has GRIC2?
Yeah, I'm sorry, I didn't catch that, Keith.
Oh, how do you? I asked how you find a mouse, but how do you make a mouse? What's the process of getting a mouse that has a human variant?
Right, so we use CRISPR-Cas9 gene editing techniques, and like many universities, we have a core facility at Northwestern that's dedicated to that technology. And they've been successful in our case for generating 2 of the GRIC2 knock-in lines. I think it's what Rick said is important, in that as researchers, we have a toolbox. None of the tools that we can use are perfect. Mice certainly are one of the tools, and then there are other approaches like iPSCs or organoids that have their own strengths and weaknesses. So when you make a mouse, you don't really know what you're going to get, whether it's going to perfectly reproduce the symptoms or the phenotypes that are seen in patients. That's probably not going to happen, but one hopes that there are enough phenotypes to make it a useful model for further preclinical studies.
I was at a conference for Fragile X a few months ago, and one of the speakers there said that we cured Fragile X in a mouse 25 years ago, but haven't been able to cure it in a person yet. So how do we know if something that will help a mouse will help a person?
I mean, I can take that. I mean, I think it really depends on the mechanisms. I think it depends a lot on the basic science. If we really, in many cases, Geoff and I have been working on AMPA receptors for 20, 30 years, and we know what they do and how they function really incredibly well, including SYNGAP also, we know exactly what it does. And so I think it's much easier to approach that. Fragile X is, I think we know a lot about what Fragile X does, but really in my mind, it's a much more complex situation. And so it's been very difficult to develop therapeutics for Fragile X, although, I think recent progress is happening. So I think that's, and really, you don't know. You develop these reagents, which we'll talk about, and when you start to go into working with biotech or getting out into the clinic, you have to test it in other animals, and especially safety issues, and try to repeat what you see in higher species or and we'll talk about the safety issues, which are really key. The bottom line is you never know until you get it into humans, and that's when you got to make sure it's really, really safe before you do that.
Correct me if I'm wrong, but my understanding is that the work that Geoff is going to do targets the DNA. The work that Rick's team has done targets the RNA. What are kind of the pros and cons of each?
Well, I'm also targeting RNA.
Oh, okay.
On the same page.
Okay. So Amy is some of her work's more focused on the DNA side, but RNA generally is something that is a shorter term intervention.
Well, I think there's lots of ways, and I think we need to try them all. So the ASO approaches that many of us are doing are regulating RNA expression, or in some ways, and eventually protein expression. As we know the ASOs are transient. That's good. So if they are detrimental, we can stop the treatment. But at the other end, we have to reapply them, every 6 months or whatever, however the stability of the ASO is. There are, lots of other approaches, genetic rescue, just rescuing with AAV delivery of the gene, and those are more long lasting. And I think there's concern, at least within the SYNGAP community, that they're a little more hesitant about AAV treatments. But AAV treatments are getting much, much better, I think, and much more safe. And then there's CRISPR approaches, which are, I think, there are different kinds of CRISPR approaches, whether it's CRISPR editing or CRISPR-A, where you regulate the transcription of genes. Those are more, again, permanent, so you can't stop them once it's started. So that's the it's one and done, so you don't have to worry about redosing. But and then I lost my train of thought there, so I'll turn it over to Geoff.
Yeah, I guess in the case of the DNA, you have to be certain that change you make, that it's going to last for the rest of the lifetime of the patient or the person, is truly beneficial, is not going to cause harm, instead of correcting or improving quality of life of that individual. And in many cases, that's just very hard to establish. Calculated risks, I guess, are taken, especially for the most severe cases where the quality of life is quite low. Then it's been justifiable from a bioethics standpoint, but that's probably not going to be the case for many genetic disorders.
Okay, thanks. Well, why don't we turn it over to?
Let me just
Go ahead. Yeah.
I regain my thought here, which is one problem with AAV genetic rescue is overexpression, and so if as we know many genes are loss of function, but if you overexpress, you can also have beyond what's normal. You can also have detrimental effects. So that's something that we have to be very careful with AAV genetic rescue.
Right. Where with something like an ASO that you've used when you go to human trials, you can start with a low dose and increase it over time if it needs to be more effective.
Yeah.
Why don't I turn it over to the two of you to tell us about your work. I'll see if we can get Amy Ramsey back. But Dr. Huganir, let's start with you.
Okay. Let me share my slides. So I'll just start. I only have 10 minutes. I'll do this quickly, but my interest, similar to what Karen Zito said earlier, really is basic science, trying to understand how the brain works and how learning and memory works and how synapses work. And over the last several decades, it's become clear that many of the genes and the proteins that we've worked on for years are discovered are mutated in intellectual disability, autism, and schizophrenia, cognitive disorders. And in retrospect, this is sort of obvious that these genes that are really critical for normal cognition, if they're disrupted or mutated, it's no surprise that they have effects on cognition. So today I'm going to, normally we work primarily on SYNGAP, a synaptic gene that some of you may be familiar with, and trying to develop the therapeutics for the kids with SYNGAP-related intellectual disability. But today I'm going to talk about unpublished work from my lab in collaboration with an ex-postdoctoral fellow of mine, Xiuling Chu, on a gain-of-function mutant in AMPA receptor, specifically an AMPA receptor GLUA1. So Xiuling was a postdoctoral fellow in my laboratory and was working on this project, and then has now gone to Taiwan, where she's has her own laboratory, and she's continued on this work, and hopefully we'll have this work published soon. So I want to disclosures. I'm a scientific co-founder and SAB member of Neuroscience Therapeutics, and founder and SAB member of Sing Therapeutics. And Sing Therapeutics is dedicated to developing therapeutics for SYNGAP1 related intellectual disability. So we don't need a lot of introduction here, but we have focused on the excitatory synapses in the brain. They're about 70% of synapses in the brain, and they work through these the GRINs and the GRICs, and so both by through NMDA receptors, AMPA receptors, and kainate receptors. So we have studied all three of them, but we have focused on AMPA receptors because they mediate the majority of synaptic transmission, active synaptic transmission in these synapses. And then, of course, over the last several decades, many laboratories have shown that synaptic plasticity, that is important for learning and memory, really is regulating the level of AMPA receptors at the synapse. So increasing strength or long-term potentiation increases the number of receptors at the synapses, as well as the properties of the receptors. And then long-term depression or decreases in synaptic strength, or decreases in synaptic function, and decreases in AMPA receptor structure or level and function. So the AMPA receptors, as well as all of the glutamate receptors, are sites of mutations, and many of these variants can produce or are involved in intellectual disability, autism and epilepsy as well. Even the AMPA receptor GLUA3 subunit has one of the most ultra rare variants associated with schizophrenia. So we began this work actually maybe I don't know, even 10 years ago, where we developed the mouse that in GLUA1 that had this very common mutation that occurs in all of these AMPA or these glutamate receptors. So this is a region shown over here. Go back. With it's conserved in GRIN2, GRIN1, GRID2. This is the famous Lurcher mutation, as well as in other subunits, AMPA receptors. And you can see over here, it's a mutation that occurs right in the pore region, and it regulates many of these mutations result in gain of function of the receptor. So we decided for a variety of reasons to make this mutant. The kids, actually I'm not up to date, but I think there are at least 12 kids who have this mutation. And they are associated with intellectual disability, autistic spectrum disorders, as well as seizures and other phenotypes as well. And so we decided to make a mutation in mice and use CRISPR-Cas9 as Geoff mentioned. And we, luckily these receptors are highly conserved between humans and mice. It's not perfect, but they're highly, the structure is highly conserved. And so we were actually able to make exactly the same mutation in the mice. And so central question is whether GRIA, this mutation is the causal mutation for ID and the autism. What are the underlying pathological mechanisms? And how can we alleviate these symptoms in the mutant mice? So we rapidly found that these mice have learning deficits. So using a classic water maze approach shown here, I won't go into detail, but it's a learning paradigm where the mouse learns within a tub of water where there's an escape tower that it can get out of the water. Mice generally don't like to swim, and so they'll find this when they swim around and bump into it. And you can see that the wild-type mice will learn and get to this platform over time with training, where this mutant mouse does not learn it. And this also affects learning, the learning 24 hours later. The wild-type mice prefer the region, the quadrant of the pool where the platform used to be, and the mutant does not. Now, we can also look at social preference. So mice are social animals. So if you put them into a chamber like this, where they have two cages, one with a mouse and one without, they prefer to interact with the one with the mouse. And this shows that this social preference shown here, where the mutation totally eliminated the social preference for the mouse. They even like novel mice. So if you put in a familiar mouse on this side and a novel mouse on this side, they'll prefer to interact with the novel mice over the mouse they already know. And you can see this in the wild-type mice, but not in the mutant mice. So they have the both learning and social preferences deficits, and we have a lot more data in the paper. It's already on bioRxiv, and hopefully it will be published soon. So we started characterizing what's going on here, and I'm skipping over tons of data. But what we found is that the as I'll show you here, these mice actually have increased calcium permeability AMPA receptors. So Derek was talking about earlier that regulating calcium permeability in these AMPA receptors could be important in the etiology of these disorders. And here we find that the synaptic transmission in these mutant mouse can be inhibited by NASPAM, which is a drug that will selectively inhibit calcium permeable AMPA receptors, where the wild type mice does not. So this is a major issue, we think, in the etiology of the mice, and actually causes a progressive neuronal loss shown here in the hippocampus. Looking at a stain that stains dying cells, you can see that in the wild type, even at early stages, there's an increase in these spots shown here at P21 and especially in the adult. So there's a selective degeneration of these, especially in the hippocampal region over time. And this is quantified over here. So this can also be seen by activation of astrocytes and microglia, as you see in the wild type versus the mutant. The mutant shows activation of these cells that respond to dying neurons. So what do you do? So what we've done and have a lot of experience in is in developing ASOs, and ASOs can be used for many different approaches, either down-regulating, in this case, trying to knock down the mutant allele, get rid of that dominant gain of function mutant receptor subunit, or you can regulate splicing, or you can regulate upregulate, increase expression of genes, and that's what we are trying to do with the SYNGAP gene. So here we developed two ASOs that are designed to target the mutation in the GLUA1 and selectively. So here, this is a gapner, which will decrease the expression or knock down the expression of the mutant allele, and you can see that looking at the mutant RNA, we have two different ASOs that decrease the expression of this, but the ASOs have no effect on the wild type gene, as shown here. And this is really critical. You don't want to be knocking down or decreasing the expression of the wild type gene. So heterozygote.
Rick, sorry to interrupt. We've given you guys a tough task of outlining in a short time, but if you can wrap up in a minute or two, that'd be great. That'd be great. Thank you.
So remarkably, these ASOs totally rescue this effect. So if we inject these ASOs within 2 days after birth, you can rescue all the behaviors as well as the degeneration as shown. Here's the degeneration. I'm not going to go through it. And here's the behavior. So here you can see remarkable rescue of this learning paradigm. So so this is just a summary, and then I just want to acknowledge the labs. So here's my lab in Baltimore. Here's Shuling's Chab lab in Taiwan. And it's been incredible to have my former postdoc continue with this line of research and really, tie all the bows and cross the T's and dot the I's on this project. And current funding, which is less than it used to be, is NINDS, NIMH, NIA, and past funding by Simons and Cure SYNGAP, which used to be called the SYNGAP Research Foundation. So I'll stop there.
Okay, thank you so much. I saw someone said they want to learn more about the rescue part of it, and so hopefully we'll have time to come back to that in the Q&A session. But Amy, are you able to go now? I know you had your computer crash, and are you ready?
Yeah, I'm so sorry. My computer did crash. Quinn, I emailed it to her and she's going to show it for me.
Awesome.
So Quinn, I'll just say next when I go to the next one. And my apologies. I hope I didn't keep you waiting. Okay, so next, please. I wanted to just Quinn, if you could go forward one. Just wanted to disclose that the work that originated from the lab has now been the foundation for forming a company, and that's what you need to do to get the second set of money to bring something from an academic endeavor to try to bring it to the to patients. So if you'll go next, I wanted to mention the 3 mouse models that we have. We're focused on GRIN1. 2 of the models are patient variant models, and one of them is the model that we've worked with the longest, which is just has low levels of expression of GRIN1. We also have developed a GRIN2A variant, and I think that we have the GRIN2B variant. We have the founders, but we haven't verified whether the founders have transmitted yet to the offspring. So anyways, we've got some new lines that we're working on as well. If you'll go to the next slide, please. Thank you. One of the reasons why gene therapy might be better for treating GRIN disorder than a small molecule is just the receptors are so complex. They need to do so many things. They need to be assembled properly. They need to be trafficked properly. The channel needs to open at the right time, close at the right time, and that allows just the right amount of ion flux to happen. And of course, there's a lot of non-ionic signaling that also needs to happen at just the right amount. So when you have just a single missense variant in GRIN1, it can be the wrong shape. That can cause that subunit to be discarded. It might cause it to get stuck in the wrong place. It might affect the gating properties. So there's too little ion flux or too much ion flux. So really, one drug is not going to be able to address all of these different problems that can happen with different variants. On the next slide, some of the things that we wanted to consider when we were developing a gene therapy was whether we should use DNA or RNA, the delivery method, whether it's a virus, a lipid nanoparticle, or a virus-like particle. There's a lot of different approaches. You can supply a gene, instructions to make the gene that might be missing or not working. You can use antisense oligonucleotides. You can use CRISPR type machinery that can cut a gene, it can edit a gene, it can make a gene express more or less. So there's a lot of different approaches that can be used. And you can also try to develop something that's going to be variant specific or something that's variant agnostic and works for lots of different variants. In addition to all of that, you have to consider things like what's the safest approach? Has it been used successfully for other diseases? Is this something that you want to do a one-time treatment, or is repeated dosing okay? What's the age of the patient that you would be treating? And then, of course, there are gene-specific considerations. You know, so GRIN1, GRIN2A, 2B, or the AMPA receptors, the kainate receptors, they all have different expression patterns and levels of expression, the cells that they're in. So all of this you have to consider when you're setting out trying to make a therapy. I'm sorry, my phone is probably wobbling. Okay, I'm going to try and set it down
Good,
on
Amy.
this broken computer. Okay. Okay, so next slide, please. Okay, so this was the strategy that we started off with. As you know, you need 2 GRIN1 subunits plus 2 GRIN2 subunits to make a functional receptor. And the problem with the patient is that one of their GRIN1 subunits is not working properly. And so I've got a little, poison sign on it because that one subunit can be enough to cause this receptor to be not functioning properly. And so what we thought is let's just try to add more functional receptor, and increase the likelihood that what gets assembled into the receptor, if we make more functional subunit, is a fully functional receptor. So next slide. I wanted to give you an idea of the timeline of when we started this, and just the process of what goes into this. So we, there was a lot of design decisions that we made about the capsid we were going to use, the promoter, and the actual sequence that we were going to use. Then we did a lot of studies in mice to look at where in the body this goes, where in the brain it goes, how much are we delivering at different doses. Then we started doing the efficacy studies, and we looked at efficacy with electrophysiology, with behavior, and we've done a lot of dosing to try and figure out what's the right dose to achieve the outcomes for both, electrophysiology, rescuing the receptor, but also, um, the different behavioral outcomes that we care about, that could model symptoms in patients. So we're hoping that by the end of this year, we'll be ready to start the manufacturing process. But I just want to let you know that manufacturing kind of goes along in parallel with safety. And these things need to be done, they can't be done at the university. They need to be done by contract research organizations, contract development manufacturing organizations. And what they will do is they will conduct all of the studies that regulatory bodies require, to ensure that the product is safe, in terms of, um, is the process working every time, doing lots of batches, do you get the same result? You have quality assurance, there's nothing wrong in this preparation. And then at the same time, once you've kind of manufactured something through this process, then you have to do all of the safety tests. You make sure that it's working in another set of hands besides ours. You do the toxicology, and you do this in both rodents and in non-human primates. And then after all of that is done, then you apply to the regulatory agencies. If they approve it, then you do a clinical trial. And then at the end of the clinical trial, you put it for regulatory review. And then once it's approved, it can go to the market. So as you can see, this is a long process, and we're about halfway through. And I'm happy to say that things are promising. So we certainly haven't given up, and we think that this is working. So next slide. Okay, this is just an example of some of the biodistribution studies that we do, and we do this with a form of the cargo that has a tag on it so that we can trace it. And we look in tissues like the heart and the liver, and we also look in different brain regions. And so this was delivered intravenously, but this virus can cross the brain, cross the blood-brain barrier, get in the brain, and we get this nice, even distribution pattern. And what we're really trying to achieve is something that looks very similar to the normal expression pattern of GRIN1. So as much as possible, we want it to be in the cells where GRIN1 is expressed, and at about the same level. So the next slide. This data, I think I showed you a couple of years ago. This was some of our first evidence that this strategy actually works. And so we put the virus directly into the brain. And then what you're looking at here are these traces showing the receptor working. Um, um, sorry, the receptor working. And you can see that in these patient variant animals, in green on the top and in purple on the bottom sections, that their NMDA receptor function is less. Thank you, I appreciate it. And then when we administer the AAV to the mice, which is the yellow there, the gold, exactly, you can see that it restores the NMDA receptor function in both of the mouse lines. And the other thing that it restores is long-term potentiation. So here, exactly, Quinn is an expert in long-term potentiation. And so she knows exactly where to point this pointer. But what you can see is that when you stimulate these brain slices, you get this jump in the response to any single stimulation after they've been through kind of a learning, an intense stimulation. And then that change remains for quite some time. So if you measure the response exactly where that pointer is 2 hours after that burst, and you compare that to what the response was before the burst, then that difference there is long-term potentiation. So you can see that that's deficit in the two mouse lines. And then when you give the AAV, you restore the LTP. So this is really exciting. It's evidence that the approach works not only to restore the receptor, but also some of the cellular functions that the receptor has. And if you go to the next slide, this is just to show some of the behavioral improvements, and we expect to see similar behavioral improvements in the patient variant lines. But here, for example, you can see that hypotonia is rescued. You can see, yes, exactly. So the red is the knockdown, and then when you give the AAV, it's the blue where it goes back to the wild type levels or almost wild type levels. And then you see locomotor activity similarly looks very similar to wild type, and is much improved from the red, and also a test of working memory here. On the right hand side, you can see that working memory that's reduced is restored with the AAV. So I know that I came in late and probably used up a lot of time. I'll just go quickly through the next slide. We're doing prime editing as well. I think I will just leave this. If you want to ask about it, I can let you know. But it is this would be something that would be lasting. It would be an edit. So if you can go very quickly to the next slide, it's a CRISPR Cas9 based approach where you are essentially making you're making a new piece of DNA, and you are putting it in exactly the right place to swap out the misspelled DNA and try to replace it with the new one. And the advantage here is that you don't get a lot of other mutations when you do this approach. So yeah, it's really exciting, but it's very preliminary. And I just wanted to, if you go forward, the next slide. Sorry, next slide, if you're able. Okay, I just wanted to mention that we did something similar with CRE-LOX, where you use a different kind of editing. And we were able to show in adult mice that doing this an editing type approach works very well. We could rescue a lot. But if you could go forward one more, I just want to show you that you don't get a full, complete rescue. And so what we found out was that the editing machinery, it needs to be able to access the DNA. And if you go forward one more time, what we found was that we got great rescue in some types of neurons, and those are the ones that are lit up in green. We saw great rescue. But if you go the next slide, when we looked at another cell type, the gold ones, we saw some of them we got rescue, but some of them we didn't get rescue. And that's where the white and the red arrows are. So the problem is that not all of the cells had the rescue. And if you go forward one more, what we found out is that those green cells, their chromatin is more open. And so that editing machinery could have access, but in the yellow cells, that chromatin is less accessible. So the point that I want to make, if you go forward one more slide, is just that Oh, I forgot to write this down. Okay, so the issue is just that we don't know how effective prime editing is going to be, and it might depend on the gene and the location of the variant. And that's really something that we're going to have to just determine experimentally. So this is where we are with our projects. And I think going forward, I just wanted to say thank you. Next slide. Thank you, first of all, to the families. Thank you to the lab. Sorry, yeah, it's good. I should go quickly so we have time to talk. And then the last slide is just a thank you to specifically the lab members and collaborators. So thank you so much. And I'm sorry again for my technical problems.
Thank you, Amy. Okay, over to you, Geoff, to talk about GRIC.
Oh, you're muted.
Sorry about that. Rookie mistake. So I'm going to tell you about a project that is at a much earlier stage than the two that you just heard about. And philosophically, it's very similar to what Rick has done with the AMP receptors, a beautiful story that he just described. And we're asking a very similar question, but in this case, with respect to GRIC2 kainate receptors. So our idea, much like the AMP receptor project, is that we are going to test the idea that RNA silencing will rescue dysfunction in mouse models of pathogenic GRIC2 variants. And so the reason why we think this could work is that suppression of GRIC2 seems to be well tolerated in both mice and in humans. Genetically, if you look at GRIC2 knockout mice, or mice that have a single allele, functioning allele, those mice don't seem to have significant behavioral phenotypes. And then pharmacologically, in people, a specific anti-seizure medication, perampanel, blocks both AMPA and kainate receptors, and that is FDA approved for use in epilepsy. So it doesn't seem like, or it seems potentially suppressing GRIC2 might be tolerated. So what we need to test that hypothesis are suitable models. And in that case, we've, towards that end, we've made 2 knock-in mice of the sort that you've heard about. These are for the 2 most common GRIC2 pathogenic variants. And then means to suppress GRIC2. And in that, we're lucky because a system or set of miRNAs have been developed already for a different application, and that is for temporal lobe epilepsy, in which the GRIC2 gluK2 containing kainate receptors seem to play a role, and that is in fact in clinical trial at the moment. So the work was inspired by these initially these 2 kids, Katie and Marley. Katie's now 20 years old. Marley's this is a relatively recent picture. They have 2 variants that are very close in position on the GRIC2 gene, but they produce very different outcomes in the kids that have those variants. And so on the 657, which is in fact the same variant that Rick discussed, but in GRIC2, you have intellectual disability and a variety of other symptoms that you can see here on the left. But I want to underscore the fact that none of the kids, and there are 10 of the kids with this variant that we know of, have seizures. In contrast, the 3 kids with the 660 variant all are quite severe in terms of their symptoms. They have profound intellectual disability. All 3 of them have to use respirators. They're not ambulatory, and all 3 have seizures. And that, again, focusing on that particular aspect of their disorders. So we generated the mice for several reasons, but one of them was to determine whether mice with the same knock-in mutations have a similar severity differential in as far as we can measure. So those mice are described here. The 657 mice, I'm not going to speak a whole lot about for the interest of time. They're not really affected in terms of their growth or viability. They do have cognitive deficits in a behavioral test similar to the one that Rick described. And they have other phenotypes, but importantly, they do not have seizures like Katie and the rest of the kids with the 657 variant. On the other hand, the T660K mice are quite severely affected by that knock-in mutation. Their viability is reduced, they're smaller, they do have seizures, and they have a lot of other features that make them somewhat more challenging to maintain as a mouse line. And so targetable phenotypes, what I mean by this is just behaviors that we can try and rescue. And so if we introduce whatever the rescue strategy is, can we see some improvement? And so one of those phenotypes, as I said, was reduced viability and growth in the 660 mice. And you can see that here in the two graphs that starting at week 3 and all the way up into adulthood, the 660 mice are smaller. In males, this is sort of the difference from wild type mice is larger than it is in females. Another set of behaviors that we can see a large difference in from wild types are in these what we call naturalistic or innate behaviors, things that mice just normally do, like groom themselves or dig in their bedding in their cages, or take these little nestlet cardboard squares and tear them up to make their little nests. And you can see in each case, the 660 and to the same, almost to the same degree, the 657 mice don't do those naturalistic behaviors to the same extent as the wild type mice. They have cognitive phenotypes. This is a different kind of cognitive task. It's called a T-maze task. And it's very similar in that the mice are supposed to learn what a correct choice is, whether they need to go right in order to get a reward or they need to go left. And in fact, they need to alternate in order to successfully do the test. And then we train those mice to reach 70% of the time making that correct choice. And so that's what it means to reach that criteria. And what we found is that the T660 mice in many cases don't ever reach their criteria. There's about a third of the mice that can never learn this task, at least over the period of time that we attempt to teach them that task. And those mice that do learn the task, if you look in D here, perform much poorer, or you could think of as forget how to do the test quicker than wild type. And in fact, if you just introduce a delay of 30 seconds, they just can't remember which way is the right way to go to get a reward. And then the third and perhaps the phenotype that we're going to focus on to the greatest degree are the seizures in these mice. And so starting at week 5, if you just move an individual mice from one cage to a different cage, those mice will start having seizures. And that increases in frequency, and the proportion of mice that have seizures to where it plateaus is about 80% of the mice have seizures when you move them from one cage to the other. And some of the mice, there's a big range. Some of them only have one seizure over the course of that 14 weeks. Some have many seizures. So say 50% of the time they're having a seizure. Other excitability phenotypes include spiking during EEG. So this is on the bottom here, you can see these spikes in the EEG recording, indicating there's a lot of abnormal activity going on in the cortex and in the hippocampus. that does not occur in the wild type or the 657 mice. So they have a range of seizure phenotypes, hyperexcitability phenotypes, and these are all targetable in the sense that we can test to see if GRIC2 suppression normalizes these behaviors and cellular properties. And last, the relative severity, I've said this already, aligns with the patient's symptoms. So Amy and Rick just went through this very quickly, or not quickly, but they did a much nicer job of describing the possible approaches. We've decided to focus on the non-selective suppression of GRIC2, and that means that both wild type and the mutant transcripts will be suppressed, in contrast to the allele-specific suppression of the AMPA receptor transcripts that you heard about. And the idea then is that just having fewer receptors, overall wild type and mutant, is going to produce a more beneficial effect than just having normal levels of mixed wild type and mutant containing kainate receptors. As I said, we're lucky in that tools have been developed already for a different purpose, and that is for suppression of seizures and temporal lobe epilepsy. And this was done by the labs of Christophe Moll and Valérie Crepel in France. They observed that overactivation of glucose-2 kainate receptors drives hyperexcitability in a number of different types of models of temporal lobe epilepsy, including in humans. And this is resected human hippocampus there on the bottom right. So we've obtained the research tools that they use to generate this data. The clinical trial is being run is by Huganir, and it's in the recruiting phase. And so the project that we plan to do and have started is to suppress GRIC2 expression in the 660 mice with the mRNA that's been developed, compare the phenotypes that I've told you a little bit about, and then answer these questions. It's an effective approach to normalizing dysfunction in GRIC2 mouse models. And also secondarily, is the timing of that suppression critical to achieve beneficial effect? And I'd like to thank the folks in my lab and some of our collaborators, and also Cure Epilepsy and Cure Grin for the support in this project. And thank you for listening.
Okay, thank you so much to Rick, Amy, and Geoff for those presentations. We have just under 10 minutes for questions. And so please put them in the chat. There's some on the chat that we can start with, some that are already here. So Christina asked, do RNA targeting therapies have to be repeated on a regular basis like a permanent medication, while DNA targeting therapies have to be done once and then they last forever? I think it's safe to say that that's generally correct. Dr. Huganir, someone asked, is this the research that showed that perampanel stopped seizures but did not have the same positive cellular and inflammation effects as ASO?
No, it's not. It's not the studies, but this is an issue that if there's a large question about whether the seizures are really the problem that can produce intellectual disability and some of the behavioral phenotypes alone, and it's not more complex developmental and plasticity effects. And so I think that many people are finding that inhibiting the seizures alone does not appear to be a critical factor. You really have to modify the circuits and the underlying plasticity and function of the synapse to get the full effect. And so I think perampanel has been very effective, and controlling seizures, of course, is a really critical thing for the kids and the parents, but I don't think it's going to lead to affecting the cognitive phenotypes that we see.
Okay, thank you. Okay, Amy, there's a few questions here related to your presentation. So one asked if it could be across GRIN1 mutations. And then another question that we can combine with it asking about if this is just a therapy for loss of function or it could also help gain of function. And I think generally we would think of an augment strategy as something for loss of function, but maybe there's something different about GRIN1, right? That might
Right,
change that.
exactly. Yes. So the Y647S variant is a really interesting one because when we look in some contexts, like the slices that I showed you, it looks like a loss of function. In other contexts, it actually looks like a gain of function. So when we did our studies in the red blood cells, we saw actually more NMDA receptor mediated current calcium flow in the Y647S. So it's a very complicated variant, and we do see improvements in that one. So I think that the idea behind the strategy, it should work for both gain and loss of function. And the reason why is that we're really, it's a stoichiometric solution where we're just adding more wild type subunit so that there's fewer variant subunits that get assembled into the receptor. So it's not a question of making more receptors because we know that there's more GRIN1, gluin1 protein in the cell than what it needs to make the receptors. And we know this because the heterozygous mice don't have much of a phenotype. We are not seeing haploinsufficient patients. So we know that there's more gluin1 than what these cells actually need. So it's just a question of when the receptor does get assembled, which subunit does it grab to put into the receptor? So yes, it should work for both.
And that was a question from a GRIA2 parent. So it's important to note that this is something specific to GRIN1, probably
Yes,
not for GRIA, and probably not even for other GRIN
right,
disorders that an augment
right.
strategy could help with gain of function patients.
I think so, yes.
Okay, Dr. Huganir, this is a great question. So you tested the ASO just after birth. Have you done any testing later?
No, that's of course is a big question, and we've been able to do that, or in the SYNGAP field, that rescuing SYNGAP expression later in life, later in the mice can rescue many of the phenotypes. But here, we just haven't had time to do that. I think we can all agree that there are windows for many of there's a developmental window, but there's also a lot of these proteins also work in adults in plasticity and learning. But I think we all can agree that the earlier kids are treated, the better. So with rapid diagnosis and rapid identification of mutations and rapid therapies as soon as possible is really what we will strive for.
Okay, thank you. And then this will probably have to be our last question, but this could be to all the speakers. What are your thoughts on effective distribution and delivery of your oligonucleotide across the brain? Are there hurdles or in distribution still present, or have your AAV LNP strategies overcome that issue?
I didn't really get to talk much about the LNP, so I just want to say that we've tested some different formulations, and we do see a little bit of differences. Some of them have better distribution profiles than others, and what we're really looking for is one that has a profile that is again mimics the endogenous, so that it would prefer to go to neurons versus astrocytes. And so what we have right now is one that can go into all of them at about equal efficiency. So again, this might be good for GRIN1. It might not work well for other cell types or other genes. The virus seems to, we're not really having a problem. So it doesn't seem to get stuck in one place. With the methods of administration that we have, we get nice widespread distribution. So that does not seem to be a big problem with our approaches.
I think with ASOs it seems like we're getting better distribution than we feared, and so a good example of this is the GRI trials at Stoke Therapeutics. They're very early stages of this, but that is a very central nervous system phenotype, but they're having effects of ASOs on those in those early clinical trials. AAVs are, there are whole companies, there are, 10 companies more that are developing AAVs that can be targeted to specific cell types very specifically and at being applied intravenously, and then the target get across the brain, expressed in only the cells that you want them to be. They're not there yet, but there's a lot of effort going into this, and so in the future, I'm very hopeful.
Geoff?
I don't have too much to add. We're at such an early stage in the GRIC field that we're hoping all these technologies will be developed by the time we get to, if we get to the point where we can test them out.
Okay, well, thank you all very much for, thank you, Amy, Geoff, and Rick for the presentations, and thanks everyone for watching and for all the great questions.